The first proposed mechanism for ALS is known as the glutamate excitotoxicity hypothesis. Be sure to check out the first video, which covers the basic anatomy of a nerve cell, helping you get familiar with the essentials so that you can follow along in this video easily. Glutamate is a neurotransmitter. A neurotransmitter is a type of molecule in the brain and body that helps nerve cells talk to each other. Think of it as a messenger that carries signals between nerve cells to communicate with one another. One of glutamate's jobs is to excite the cell, which means that it will help make the inside of the cell more positive. It does this by increasing calcium, a positively charged molecule in the cell. Glutamate is very important, but if there's too much of it, it can overexcite a cell in a toxic way, which might cause cell damage and even cell death. That, in essence, is what the glutamate excitotoxicity hypothesis is about. But let's take a deeper dive into understanding why this happens, how it's related to ALS, and if researchers have been able to come up with a treatment for it. The problem with too much glutamate is not glutamate itself, but that it allows too many calcium ions into the cell. Glutamate is able to increase calcium in the cell in two main ways. First is by directly binding to and activating two particular glutamate receptors, AMPA and NMDA receptors. The second is by indirectly causing the opening of something called a voltage-gated calcium channel. This is essentially a protein channel on the membrane of your cell, similar to that we see for glutamate. The difference lies in what makes the channel open. Your neurons have a more negative charge inside the cell compared to the outside. In other words, it has a different voltage. So voltage-gated channels will open in response to differences in this voltage. But in this video, we are going to focus on glutamate receptors, particularly AMPA receptors. Motor neurons have characteristics that make them more vulnerable to excitotoxicity compared to other cells in the body. One characteristic is that their internal calcium levels rise very fast compared to other cells in response to glutamate stimulation. In addition, motor neurons have low intracellular calcium buffering capacity, which is something I'll describe later in the video. There are two ways that calcium will enter the cell, either through voltage-gated calcium channels in response to glutamate stimulation, or they can enter through the AMPA receptor itself. AMPA receptor is a tetrameric receptor complex, which means that it's composed of 4 different subunits. These subunits are called GLU-R subunits and are numbered through 1 to 4. GLU-R2 subunit is of important interest. It is this subunit that determines if the glutamate receptor is permeable or impermeable to calcium. The way it decides this has to do with how the subunit was originally made. But to understand this, I'll have to remind you about how proteins are created. The blueprints for all of our cells are contained within our DNA. In order to make proteins, which this receptor and its subunits are, a copy of the DNA recipe for a particular protein is made in the form of a molecule called mRNA. The mRNA is read and used to add together tiny building blocks called amino acids in a specific order. These amino acids are then linked together like a chain to form a protein. Now, if the mRNA for the GLU-R2 subunit had an amino acid called arginine, which is positively charged, added to the 586th position of the mRNA, instead of an amino acid called glutamine, which has a neutral charge, and the positive charge will cause repulsion with the positively charged calcium in the receptor channel. To provide clear evidence that this was indeed the case, researchers added more calcium-permeable GLU-R2 receptors in mice, which led to a late onset of motor neuron damage followed by a decline in motor function, similar to that seen in patients with ALS. Researchers also did an experiment with mice lacking ADAR2, which is an enzyme that adds arginine instead of glutamine to the mRNA. These mice had motor neuron damage, leading to seizures and eventually young death. But by introducing the correct GLU-R2 receptors, this motor neuron damage can be completely rescued. I previously mentioned that motor neurons have low intracellular calcium buffering capacity. What does this mean? It means that the cell has a limited ability to handle and stabilize calcium ions within it, particularly parvalbumin and calbindin D28K, making them highly vulnerable to excessive calcium influx. Researchers found that motor neurons vulnerable to ALS, such as those in your spinal cord, lack detectable levels of parvalbumin and calbindin D28K. This leads to a lower calcium buffering capacity, while less vulnerable motor neurons, like those in the oculomotor nucleus, which is just a small cluster of nerve cells in an area of the brain called the brainstem, do express these proteins and have higher calcium buffering capacities. So the question is, were researchers able to find a treatment based off this hypothesis? The first goal of researchers was to reduce the amount of glutamate released by the neuron. To do this, researchers caused a reduction in the structure that transports glutamate in the cell called vesicular glutamate transporter 2 in the neurons surrounding the motor neurons in the spinal cord. Although the amount of motor neuron damage was decreased, unfortunately neither disease onset nor the lifespan of the ALS animals was affected. The second goal is to directly block AMPA receptors. Treatment with these blockers significantly delayed the onset of ALS and increased the survival of animals. Pelampanol, a molecule that blocks AMPA, was tested in a clinical trial, but unfortunately the results were negative. Lastly, researchers wanted to find a treatment for the calcium buffering capacity problem. Researchers created genetically modified mice to express high levels of calcium buffering proteins. These mice developed less severe motor neuron damage and lived longer than ALS mice without increased expression of calcium buffering proteins. However, drugs that induce these proteins are not currently available. There are currently no treatments to stop the progression of ALS. However, there are now 3 available drugs that provide some benefits to people living with ALS. These are riluzole, edaravone, also known as Radicava, and tofersen. While riluzole and edaravone can be used by all people with ALS, tofersen can only be used by people with SOD1-related genetic ALS. Riluzole became the first drug approved in the US for treatment of ALS in 1995. As we discussed, excitotoxicity is due to an excessive amount of glutamate stimulation, and the drug riluzole tries to fix that. It reduces the level of glutamate by blocking its release from nerve terminals. Unfortunately, the effects of riluzole have only been shown to prolong survival by about 2 to 3 months. This was a small insight into the excitotoxic hypothesis for the mechanism of ALS. While efforts to reduce glutamate release, block AMPA receptors, and enhance calcium buffering capacity have shown promise, they haven't led yet to a cure for ALS. Ongoing research continues to explore these pathways with the hope of developing effective treatments. This hypothesis represents one piece of the puzzle in understanding ALS, emphasizing how complex and challenging this disease is to treat and underscoring the importance of continued investigation into its underlying mechanisms.